What is CRISPR? CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeat. This name refers to the unique organization of short, partially palindromic repeated DNA sequences found in the genomes of bacteria and other microorganisms. While seemingly innocuous, CRISPR sequences are a crucial component of the immune system of these simple life forms. The immune system is responsible for protecting an organism's health and well-being. Just like us, bacterial cells can be invaded by viruses, which are small infectious agents. If a viral infection threatens a bacterial cell, the CRISPR immune system can thwart the attack by destroying the genome of the invading virus. The genome of the virus includes genetic material that is necessary for the virus to continue replicating. Thus, by destroying the viral genome, the CRISPR immune system protects bacteria from ongoing viral infection. This technology can be used to copy and identify certain patterns in DNA and in RNA in most other organisms, and it can be used to slice and cut certain aspects of a person's or thing's genome. I started at the basics of what CRISPR is because I do not believe that this subject can be truly grasped without starting at the beginning. But I think it's important to understand how CRISPR works in its works in its own way, so that we can understand how and what it can do for human and other DNA that we think we can use this with. It is very important that we have a firm understanding of what can be done and how it is achieved. All right, what I am doing now is I am explaining how the CRISPR immune system starts and how it works at the beginning level and what it does for bacteria. CRISPR was actually discovered by scientists who were studying E. coli, and that is how we were able to come this far, because it started with bacteria, and that's why it's important to know from whence it came. The steps of CRISPR immunity. I'm going to start with the first steps. The first step is the virus will invade the bacterial cell, and these regions are composed of short DNA repeats and spacers. When a previously unseen virus infects a bacterium, a new spacer derived from the virus is incorporated amongst existing spacers. The CRISPR sequence is transcribed and processed to generate short CRISPR RNA molecules. The CRISPR RNA associates with and guides bacterial molecular machinery to a matching target sequence in the invading virus. The molecular machinery cuts up and destroys the invading viral genome. With CRISPR, we can design and build a certain segment of DNA or RNA that we can match with another strand, and we can use the strand that we designed to cut or snip away certain parts of DNA or RNA that we do not want to be there, and it can be replaced with what we have designed. These spacers that are replaced serve as a genetic memory of previous infections. If another infection by the same virus should occur, the CRISPR defense system will cut up any DNA sequence matching the spacer sequence, and thus protect the bacterium from viral attack. If a previously unseen virus attacks, a new spacer is made and is just added to the chain of spacers, then repeats. This discovery has led to a higher potential of scientists being able to identify and cut out certain parts of DNA. And there are some other steps at how the CRISPR immune system works, and they are: step 1, adaptation. The DNA from an invading virus is processed into short segments that are inserted into the CRISPR sequence as new spacers. Step 2 is going to be the production of CRISPR RNA. CRISPR repeats and spacers in the bacterial DNA undergo transcription, the process of copying DNA into RNA. Unlike the double-chain helix structure of DNA, the resulting RNA is a single-chain molecule. This RNA chain is cut into short pieces called CRISPR RNAs. Step 3 is going to be targeting. The targeting of CRISPR RNAs guide bacterial molecular machinery to destroy the viral material. Because CRISPR RNA sequences are copied from the viral DNA sequences acquired during adaptation, they are exact matches to the viral genome and thus serve as excellent guides. The specificity of CRISPR-based immunity is recognized in destroying invading viruses is not unusual for bacteria. Creative applications of this primitive yet elegant defense system have emerged in disciplines as diverse as industry,